And now it's my pleasure to introduce our next speaker for the day, Dr. Christine Diepenbrock. Dr. Diepenbrock is an assistant professor in the Department of Plant Sciences at the University of California, Davis. Her research program aims to understand and improve the nutritional quality and abiotic stress resilience of staple and specialty crops to ensure a food supply sufficient in quantity for the long term. Her research group focuses on the statistical, genetics, computational genomics, phenotyping, and development of integrative models for crop productivity, agronomics, and nutritional quality in the presence of abiotic stressors such as drought and/or high temperature. With a focus on product delivery, the ultimate goal of her research program is to contribute to the improved nutritional status of human beings and livestock on a regional and global scale. Today, she will describe a few of her group's current projects, including as part of collaborative teams that are relevant to the theme of our symposium. The title of her talk is Digital and Genomics-enabled Technologies to Improve Crop Productivity and Quality Under Abiotic Stress Conditions. So without further ado, let's hear it from Dr. Diepenbrock. You should be able to share your screen, Dr. Diepenbrock.
Great. Are you seeing the full slide?
Yes, we can.
Okay, great. Thanks very much, Swati, for that introduction. And many thanks to the UF Plant Science Council and to Jason Rauscher and team for their support and organization of this symposium. Swati already mentioned this title. I will be focused today on our group's work, specifically on abiotic stress tolerance. But our lab was established 2.5 years ago and thought to give a bit of a quick overview on one slide only of the full scope of what we're up to. Okay. Great. So the areas of focus in our lab, our overall theme and the core of our work is to improve crop nutritional quality and abiotic stress tolerance in tandem. This is due primarily to the partial overlap in target environments for improvement of these two trait sets. So many of the environments in which we're working on biofortified or nutritionally improved crops through breeding and/or agronomic strategies, there also tend to be one or more endemic stressors, such as drought or high temperatures. So the way that our work ends up looking on a day-to-day basis, we conduct quite a bit of phenotyping of nutritional quality traits. We do genomic dissection. I am trained as a plant breeder and quantitative geneticist. And then we do quite a lot with actually using that information in the breeding process in a genomics-enabled way. We also look at leveraging win-win scenarios. So it may be that nutritional quality is not yet explicitly valued along the supply and demand stream. For various crops. This can be the case for both staple and specialty crops. So we end up looking for other traits that are already highly valued that may be able to be improved alongside nutritional quality, so tandem gains there. We also are working on making sure that the nutrition that we're breeding into the crop actually makes it to the consumer. So rather than, typically it's really helpful for genetic genomic studies to halt enzymatic activity right at time of harvest, get those samples into a -80 freezer. However, most of us would not have one of those in our dwelling place. So we end up wanting to make sure that through that post-harvest, through the storage and processing phases, and including in the digestive process, we actually are providing improved nutrition all the way through to that time of consumption. And we have some interesting simulated gastrointestinal digestion projects with a food science and technology collaborator here on campus, but that is outside of the scope of today's talk for sure. And then we are doing quite a bit of work with two collaborators, one who specializes in 3D biophysical modeling and one who specializes in AI-enabled sensing, and getting those integrated into how we do molecular breeding. And finally, quite pertinent to today's talk as well, we are working on integrating physiology and genomics, focusing on food crops, as we do in all of our work, given the scope of our lab. And we are looking primarily at drought and high temperatures and trying to get submodules for crop compositional or nutritional quality traits integrated into some of those crop growth modeling frameworks, be they mechanistic, 3D, or otherwise. Oh, I see a hand raise. Hopefully Swati will see. I'm glad to stop if needed. Okay. So for an overview of what we'll talk about today, again, these are the 4 projects in our lab that are most pertinent to the theme of this symposium. I will say this is a bit of an unconventional talk for me. You know, we tend to, nearly all of our projects focus on nutritional quality, and then some of them also have an abiotic stress component. So to pull out specifically our abiotic stress side of the work was an interesting exercise for me, and hopefully it's an interesting and informative story as part of this symposium content. So first I will describe an experiment that we recently finished where we integrated mechanistic crop growth models and whole genome prediction in a maize breeding experiment. These crop growth models often operate on a daily timescale and would be somewhat akin to those described in the platform that Anju was just speaking of. Notably, Dr. Charlie Messina, who I understand has joined you all there, was a critical collaborator and the senior author on this work. So feel free to ask Charlie any questions in addition to any that you'd like to send my way. Second, we have some work in Southeast Zimbabwe that I'll describe also in maize, and then some work here in California and South Africa in sorghum, and finally that AI-enabled sensing and modeling. So we'll move through those, and then just one slide at the end with some concluding thoughts that overarch all of those four. Okay, so integrating crop growth models and whole genome prediction. Some of you all in various crops may be working with whole genome prediction and or genomic selection. But in this case, we wanted to see what is the utility of integrating these crop growth models. And we just had this paper published last year in Plant Physiology. And feel free to check that out, but I'll show a couple of the figures and main findings here. Great. So here's the kind of framework of CGM-WGP. And this is a quote from Charlie et al. A few years ago. So this is a generalized linear model version of WGP, where the crop model acts as a link function. So effectively in standard genomic prediction or selection for yield, what we might do is fit marker effects for yield and then try to predict breeding values for yield. In this case, what we do is we actually have a crop growth model in the middle. So we could end up having observed yield from some number of field environments. We can run this crop growth model backwards to estimate physiological traits at the level of individual genotypes, which might be helpful for us as breeders. We then get those regularized to marker effects, which allows us conveniently to then predict physiological trait values for tested or untested individuals. And then this crop growth model provides a way to integrate the environment and management information, so such as the weather and soil data that were just described in a recent talk as well, as long with irrigation and any other factors that might be important. Once we have done that, we can run this model forward for tested or untested genotypes, tested or untested environments, provided that the assumptions of the CGM are still reasonably met for those E and M conditions. And move this model forward and get predicted yield. So that's kind of the cycle of how this works. There's a whole lot of sampling that goes on. This is a Bayesian framework, as you might notice with these priors going on. But yeah, I'll just jump ahead and show you what's going on here. So you're getting the most detailed version. There's also a summary slide next. But effectively, what we did was we compared CGM-WGP predictive ability with that of classical WGP, in this case represented by a method called Bayes-A. So we looked at all four quadrants of prediction, tested and untested genotypes, tested and untested environments. And on this left side, we looked if we only included well-watered data. So these are primarily from the target population of environments for corn throughout the US Corn Belt. We had several sites across a couple of years. And we found that the CGM-WGP method was performing at parity or slightly better than, in the case of this one quadrant, WGP in each of those four quadrants of prediction. In somewhat contrast, if we start to include data from managed stress environments, in this case, this would be flowering stress, which would be a critical period for maize to experience drought stress. So we end up seeing in that case that the predictive ability of CGM-WGP is maintained relative to only having well-watered data in the model. So if you compare each of this, say 0.79 to 0.8, 0.61 to 0.58, et cetera, whereas predictive ability of WGP, once adding in that managed stress data, really does drop off. So from 0.72 to 0.49, 0.61 to 0.42, et cetera. So the CGM-WGP was able to maintain its predictive ability when integrating that information. It also was able to, even in non-stress, relative non-stress conditions or relative absence of G by E by M, we end up seeing that there's not really a penalty to having CGM-WGP or having a crop growth model in the model, or in the, excuse me. It doesn't hurt to have a crop growth model in the middle, effectively. These performances are still at parity. So we do see a greater advantage in this situation. We saw that both across family, which is represented up here, and within family. A lot of these points where CGM-WGP was outperforming WGP was in the case of these managed stress environments in particular. And the main goal of these typically would be to test the effects of a given stressor and test material under that stressor in a manner or in a place that can more consistently apply that stress. So it may be that in the target population of environments, you may only see drought, say once in a decade. This was the case in 2012 in maize, quite badly in parts of the Corn Belt. Whereas these water deficit environments were in California and Chile, and are able to reliably apply a stressor and give us that data even in a year that wouldn't be a drought year in the target population of environments itself. Okay. Great, so here's the text summary in case you prefer this, this version rather than looking at lots of quadrants of things. So effectively, we do have this performance at parity or better in all 4 quadrants. And we also notably saw that this CGM-WGP method, we were able to stably estimate multiple physiological traits. In this case, a couple relevant for yield potential and a couple relevant for drought tolerance. And this was the case when we included multiple environment types in the training set, so both the well-watered and the managed stress environments, and when we were estimating multiple physiological traits simultaneously. So we didn't start to get combinations that really threw the model off or were not biologically feasible, et cetera. So this was promising in terms of the method stability and reliability. Here's a schematic of how effectively which parameters we might pull out and how they get estimated. So here is a schematic of a crop growth model that would again be run on a daily timescale in this case. So we start over on the left, we see various sub-processes relevant to plant growth and development. We would start out in the vegetative phase, of course, and then move into at some point the reproductive phase to start to accumulate ear biomass and eventually get to your yield and harvest index. So for any one of these sub-processes, there's been various really intensive physiological experiments to get some of these equations and parameters established. And in this case, what we could do is take an equation for radiation use efficiency, or biomass accumulated per unit intercepted radiation. We could pull out a certain parameter in that equation, and then we could allow that to be estimated on the basis of individual genotypes, and then get that back to marker effects. So that's an example of one of the types of parameters we could pull out as this maize plant is growing and developing. One of the opportunities I'm really excited about with this type of work and this methodology is that we can start to look, so this gray cloud is effectively the germplasm space in this entire population that we were examining. And in this case, we can look at certain crosses and look to see effectively where the progeny of that cross fall out in physiological trait space. So in this case, if we cross these two fairly elite parents, we end up seeing very high scores for senescence, which actually is disfavorable for stay green. So the progeny of this cross are tending to not have the stay green trait. And then we can start to look at, as we make different crosses with various drought tolerant parents, again, how that kind of progeny cloud moves around in physiological trait space, which it seems is really interesting in the case of, can we look at which strengths of parents might be redundant or helpful and complementary in some way, which vulnerabilities might be exacerbated or ameliorated. So this has been a pretty neat method to be able to look at how these traits and yield, of course, are related to each other as well. This would be a principal component analysis by plot. And yeah, we could do this. We could do this in any number of ways to slice this data, but this ends up being an interesting one to see what trait combinations we have, and which crosses we might think about making that might end up producing largely overlapping clouds. And maybe that's a good thing if we only want one tiny part of this cloud. But if for parts of our our TPE we need some physiological diversity embedded, we might need to think about which progeny for which set of environments, including in this space. Okay. So that's it for that part. I'm planning on leaving plenty of time for questions. So we're going to jump around a little bit, and hopefully feel free to write down or save your question, or go ahead and put it in the chat or Q&A now as well. So we'll just continue on since this one's also in maize. This is a partnership with the International Maize and Wheat Improvement Center, and the way this one came about is that as part of my PhD, I ended up working extensively on carotenoids and tocochromonols. So some carotenoids have provitamin A activity, some tocochromonols have vitamin E activity, and these are lipid-soluble antioxidants. So I was looking at those sets of nutrients in largely temperate panels. However, biofortified maize has typically been being developed in the tropics and subtropics. So ended up, I did a graduate research stay at Zimbabwe, where we ended up evaluating a diverse hybrid panel, yellow to orange grain here, meaning that this is non-negligible accumulation of carotenoids in the grain, and wanted to look at how those lines performed under drought and high temperature conditions. So to give a little bit of a background on the status of orange maize, there have been varietal releases about a decade now. They started in Zambia and have expanded in various ways. And there end up being a mix of varietal types that are available, depending on the agronomic context and existing farmer preferences where they are being developed. However, agronomic viability, namely tolerance to abiotic stressors, has been a limiting factor, including as some varieties are moved around in a somewhat regional way. There end up being at both the inbred and hybrid level a need to improve that productivity. The inbred level, of course, for purposes of seed production in the case of hybrid varieties. And finally, we did have the case where yellow maize ends up being dispreferred in some regions. Our group has some separate studies that we've now completed where we've dissected the genetic basis of carotenoids and kernel color in tandem, which was an interesting study to look at that element of consumer preference and nutritional quality at the same time. But that is, again, outside the scope of today. So I'll just talk about the part of this study that had the stress component. Okay, so there end up being quite a few on-farm trials, and here's the products of orange and the white-grained maize side by side. Quite a few breeding lines and effectively snacks produced from orange maize to show how that color permeates through the different food products. So again, our goal was to look at carotenoid-dense hybrids and identify those with high yield and other favorable agronomic properties that are critical for adoption effectively by farmers and farmer preferred under combined drought and heat versus high temperatures, combined drought and high temperatures here on the left, and high temperatures only well watered here on the right. So we had three trials in total and have been analyzing those data. We've seen some pretty interesting relationships between some yield and some of the agronomic traits, which could be informative for selection. And our repeatabilities and heritabilities looked really good in both treatments, thankfully. So that was good news. Okay. And then finally, again, we're getting back into genomics-enabled breeding strategies where we're working on identification of favorable alleles, development of GP models, which look reasonable so far. Not as good as they often do for nutritional traits, but there's various factors that can go into that. And finally, some candidate germplasm. Okay. So yeah, this was a 350 line hybrid panel selected for various reasons, crossed to a common tester, which is a common practice in maize. And we ended up testing in Southeast Zimbabwe, where these drought and high temperature stresses, again, are endemic, and yield loss can be quite severe. So here's that population kind of laid out. So I won't go further into detail on that panel at this point, but it's been a really interesting collaboration and a great chance to kind of go into the target environments and make sure that, we have a lot of work that we've done in largely temperate panels, but take a look at some of these relationships in tropical, subtropical material. And we're working on some of the nutritional and consumer side in that material now as well. Okay, we'll keep moving. So this one is probably the second largest example out of the 4 that I'll show you. I'm just checking time. Okay. So we do have a project that is funded by the USDA NIFA Foundation on Applied Science program, where we are dissecting productivity and quality traits in tandem in grain sorghum. So we again have a managed stress treatment with pre-flowering drought, and we also have a well-watered treatment. So these two are side by side. This is a partnership with Dr. Sibiya, who's a maize and sorghum breeder at the University of KwaZulu-Natal and the African Center for Crop Improvement. So the way this partnership came about is that we both had interest in grain compositional nutritional quality traits. Pre-flowering drought stress was relevant in both sets of environments. And then we had an interesting, again, the germplasm became important. We're testing a diversity panel here that has all 5 working groups of sorghum represented, whereas production in South Africa is focused primarily on kefir lines. So we end up having parallel germplasm sets that are of interest in each place, and then applying some set of co-developed methodologies. Okay, so here's what we're up to. We have 2 field seasons now done, and had some pretty good, pretty interesting stress responses that we exhibited, which I'll show you shortly. We are doing nutritional phenotyping, macronutrients, mineral nutrients, and we're looking at lysine, which tends to be, it's an amino acid that tends to be limiting in cereals. CIMMYT had quite a good success developing a near-infrared spectroscopy method for quantification of lysine as part of their quality protein maize efforts. So we're going to see if we can get that up and working in sorghum. And then we'll do GWAS, or genome-wide association studies, and then we'll do either genomic selection and/or marker-assisted selection as looks appropriate for the traits as we analyze them for both of these sets of traits, and then we'll test this crop growth modeling component and try to start to integrate a macronutrient compositional module into that framework. There's a little bit of linkage already present that we can build upon. So we'll see kind of how standalone versus integrated that module ends up being, but that is a direction for us going forward that we're taking. Yeah, we're going through the first phase of that now. Great. So why would we look at pre-flowering drought in sorghum? In this case, that developmental window coincides with the determination of grain number. So there's great work from Rick Boyles and team who have found effectively, they found grain number to potentially be more susceptible to GE and M rather than grain size, and also that this grain number ended up corresponding more closely with final yield than grain size. So it looks like an important one. It looks like it might be more susceptible to the fuller set of GE and M main and interaction effects. It's a great paper if anyone's interested. Okay, cool. And then why would we look at grain compositional traits? Again, as mentioned, there's a lot about grain composition and nutritional quality that A, is not incorporated in crop growth models, and B, that is not known or less studied even in well-watered conditions, much less under stress. So we're looking at the impact of, in this case, stress while the grain number is being determined on later season phenotypes, which end up relating to various pre-flowering processes as well. And then finally, we do have some valuation of grain composition along the supply and demand stream in this case. And sometimes the trait preferences are in different directions across these different use cases, and sometimes they're in the same direction. For instance, protein digestibility might be helpful for multiple of these. So we have a variety of selection targets, and they may, in some case, we may end up needing to develop divergent populations depending on the final intended use. Great, so here's the panel that we are using, and we are testing, Davis is just west of Sacramento, where I am currently. We're testing about 3 hours south of there, about an hour west of Fresno, if that's helpful. Rebecca Dumansky is the PhD candidate carrying out this work in our lab. And we have, again, 2 seasons now. Both have been during a pandemic, which has been an interesting time to start field trials in a new place. But hopefully that has been okay for you all as well, if you were conducting that kind of work. So we do end up seeing, in the well-watered treatment, we do see quite a higher skewed distribution of yield, whereas we do see that penalty imposed in the pre-flowering drought treatment. Similarly, we see plant height suffered in that pre-flowering drought treatment, and flowering time ended up being substantially later and somewhat of a wider distribution. I'll come back to this point at the end of this talk. We did use an augmented design where we had 2 master checks in each of our block of 15 plots. We ended up, for logistical reasons, needing to use a different check this year, but there's been some interesting work in the breeding space with regards to the use of repeated checks and kind of what they enabled you to do statistically. But this decision was more logistical than anything in this case. And we ended up seeing pretty good repeatability across reps within treatment. This is from just the 2020 data so far. At this point, we're still working on the 2021. So in pre-flowering, again, we saw pretty good repeatability there. This is across 2 replicates within a treatment for each of these agronomic traits that were assayed, and then slightly higher in the well-watered, which I guess would not be too surprising. But that's a whole nother debate I won't get into. Okay, great. And then we did actually see quicker flowering time in 2021 in both treatments. So I guess flowering sooner than this 1:1 line in 2021 relative to the time that these plants flowered in 2020. We did have quite a hot year. We also had less wildfire smoke. And with only 2 environments, it'll be hard to distinguish exact effects of those 2 in terms of specific environmental covariates, but we did see a faster season this year up through flowering. Then it cooled down, so I guess you never quite know what'll happen. So some of our initial results for looking at composition, I thought to just include a couple of slides of how we go about doing this. We ended up, we've looked at a set of 40 plots so far, and we have hundreds we've been closed again due to Omicron for a lot of our research ops, but we'll be back online starting next week. So we'll be getting through the larger portion of our trial very soon, which will be great, where we have already starting to see a range in protein, even in 40 plots, and we have thousands of plots. So we're expecting that these ranges will broaden even further as we start to look at the full panel. Ash ends up being total mineral content, which can be useful for different use cases, and is also very easy to assay alongside these other traits. We've ended up looking at how well our pre-made calibrations are working on this instrument, which sits on the benchtop. And in ours, in this case, it includes visible wavelengths, but goes through a wide range of wavelengths, and you end up being able to predict different traits based on their spectra. So in this case, we already have a model that's working pretty well for protein prediction, reasonably well for total mineral content, and then we're going to need to do some work for these other traits. some of which just aren't included in existing calibrations. So we'll get this up and working. Okay, and I'll mention R-squared isn't everything, as some of you might have run into, but we have various other metrics that indicate model and calibration performance in the NIR space, which are extremely helpful to us. Okay, I mentioned lysine already. Yeah, seed storage proteins tend to be deficient, like characteristically deficient in lysine. So we want to make sure that if we're increasing protein content, we're also paying attention to protein quality. So that's one of the reasons why we're looking at lysine in addition to just protein content as a percentage of grain weight. And then we're also looking at total starch and total phenolics. Okay, and finally, look at time, great. So we also have a project that is based out of an AI Institute for Food Systems here on campus. And this is led out of UC Davis, but has a number of partners. And we are our lab is participating in the molecular breeding cluster of this institute among 5 other clusters that exist. So this is our year 1 project, and we've slightly shifted in year 2 what we'll be doing, but we'll describe kind of the points that are in common as we have progressed in that work. So what has been our challenge is that we want to be able to effectively integrate between sensing information and some of our ground truth information, and eventually integrate with genomic data as well. So we do want non-destructive sensing. That's helpful because we need the plants to still be able to be harvested at the end, especially when we're also working on nutritional quality and we need those samples. We also want simultaneous sensing of as many traits as possible, so we aren't having to deploy several different platforms. And we are really interested, and this is another way to summarize our group's work, is that we would like to be helping to integrate biochemical and physiological traits into the breeding process in a routine and cost-effective manner. So this is just another way of carrying out that objective for us. And we really want to enable selection. So this provides information on individual genotypes. It provides information that is readily integrated with some of the current data streams that we're already using in our breeding. And finally, we are looking at a crop growth and development component. So we'll keep trying mechanistic models. We're also testing some AI-enabled models that may be less mechanistic, may not operate on that explicit daily timescale. And we'll test some ensembling of those. It's pretty cool. Leafy greens are an interesting example. It's a very different crop structure than those that we've worked on so far. And then in grain legumes, we're just kind of focused on broad scale structural properties rather than trying to build up the whole plant part by part. So we'll kind of test out these simple methods first and then slowly become more complex as needed. Again, we are targeting both yield and quality. This is something we try to do whenever possible. It is experimentally complex from a logistical perspective to do so, because you need to get good yield estimates and you also need to get good grain samples. So we've explored different ways to do that, and a small plot research combine is really helpful. But yeah, it can be a challenge to try to do both trait sets compared to only focusing on one. We do have quite a bit of sensing data on various crops. We have a different suite of data available. We of course have our destructive ground truth, which we know is imperfect, but have ways to thankfully look at repeatability and that type of thing, as we might tend to do as breeders anyway. We have quite a bit of field experimental metadata, and this has been an interesting feature of this project, given that we're collaborating with a few different breeding programs. So it's been a neat experience to kind of familiarize with the different designs that the groups are using and different kind of aspects and relevant features of their germplasm. And finally, we're working on genomic integration of genomic data. We're leaving transcriptomics alone for now. Should have taken that out. Great, so this does involve some convolutional neural nets, and this is thanks to a collaborator that I'll show on the next slide. We're working on some 3D biophysical modeling, which again, I'll show on the next slide. And then finally, this last part is going to be more similar to what we've been doing with genetic genomics, but we'll integrate those sensing data streams. So there's some interesting looking methods for integration of hyperspectral and genomic data, or various types of sensing data with genomics, and various, effectively, there's so many arrows that can be drawn that we're testing out which ones might make the most sense and be most efficient. Okay, so this is a great team that we've been able to put together for this project. It has taken some pretty interesting combinations of skill sets and interests to get this team together, but Johnny and Earl have been carrying out great work and are making sense of a lot of the sensing data and in all of its complexity, which has been great, and they have really relevant experience. So what we're working with on the prediction and modeling, so AI-enabled prediction, this is that multi-input, multi-output CNN strategy that I mentioned, and we're working with Mason Earl's group on that part. They have some interesting papers and preprints out if you're interested in sensing and would like to look them up. And also with Brian Bailey, who is in our department in plant sciences. And here's an example from Brian's paper, shared with permission, where they, so here's a Stanford dragon, which I guess is a classical object to model, and then a few examples in crops. One example in which 3D modeling might potentially be helpful, and this is a problem that Mason and Brian have been dealing with, is the matter of occlusion, where you may not be able to see everything that you're interested in sensing or modeling. So one example here that I've depicted is the crown of a common bean plant, where it may be that those beans are somewhat in the understory, and it might be harder to visualize them from top view or even potentially side view cameras or other sensing devices. So how can we work with 3D models in a scenario that involves extensive occlusion for our traits of interest is something that we've been working on, and thankfully Mason and Brian have some really interesting results thus far. Okay. Great, and then I'll mention also with Mason and Brian, we are working on explicitly integrating all 3 of these domains. So our group is doing the pre-breeding genomics side, and Mason is leading the AI-enabled sensing, and Brian leading 3D biophysical modeling. There is a press release that was just put out with regards to this project. There's a student article that's fantastic that's coming out today, but I haven't seen it posted yet. Maybe by the networking session tonight. Feel free to reach out if you're interested. It's kind of a longer form description of what we'll be up to. I'm really excited about this project. I would note that URLs do not allow decimal points, it seems, so we were not in fact awarded this amount, but it is a fairly large effort, and we're very excited to pursue it. It's been a neat process starting to sort out how we can familiarize with these different domains and integrate them, any set of 2 or all 3 of them. Okay, so I just thought to give a couple concluding thoughts, particularly given that I just talked about 4 different projects. So we effectively, abiotic stress tolerance is showing up quite pervasively throughout our work. Again, that's part of why we focus on abiotic stress tolerance in tandem with nutritional quality. We are seeing this show up for staple and specialty crops. Our group does work on both, and we're seeing this show up within and across geographies. So in our work in California, abiotic stress tolerance has of course been huge. And in our work in parts of Southern, East, and West Africa through collaborative partnerships, again, this has become a major focus for us. I would note that when we're carrying out when we're carrying out managed stress trials, this does end up to have, we typically end up having fairly large experiments, especially if we want to have that control treatment, which is very helpful to us and I would strongly advocate for. But effectively, if you would like to have both a managed stress treatment and a control treatment, that ends up being a large field. Especially if you want to test combined stressors. Again, it's really important to consider, of course, all our dimensionality of how many genotypes we're able to test in how many environments, and not spread too thin. However, these trials are immensely important, and they were really helpful in that CGM-WGP context. And they also give us a really important comparison of and look at what we might be facing coming forward as climates are changing, and as endemic stressors are already faced and have been for years and years. Also, I would just say this importance of testing in the target environments whenever possible, through partnerships if you're not actually based in that environment, and letting those partnerships lead and drive the agenda, of course. And also knowing the germplasm. So in our case, we were looking at panels that were largely temperate in their adaptation, or up to 25% tropical, or in one case we had a nested population of which about half the parents were tropical. But it's been really interesting, including in a prediction context, to consider the adaptation of the germplasm, especially if we're relying upon relatedness and making our predictions. Thinking about how we can bridge those populations ends up being a really important and interesting consideration. Finally, I'll mention, we've mentioned some of these modeling and sensing techniques that we're integrating. Strategically expanding to bring in more toolkits can be really helpful in addressing our major research questions of importance for improved crop productivity and quality. It does take time to expand and include these toolkits, and I would say collaboration is absolutely key, both in terms of those target environment partnerships, those methodological partnerships, the interdisciplinary teams that it takes to look at abiotic stress tolerance, number one, and also integrate some of this physiological insight and knowledge into our genomic strategies. So although I'm biased, I would say integrate physiology into genomics, whereas others might say integrate genomics into physiology. So it certainly goes both ways and then some. Okay, I think that's it. Many people to acknowledge. So here's the full CGM-WGP co-author team. Again, Charlie Messina is there now at Florida, and hopefully feel free to sit down with them and have a chat, although I don't want to volunteer them, but I'm sure they might be excited to talk, and I am as well. I'm glad to anytime. That Zimbabwe team, Jill Kearns is a physiologist there. Toko and Leila is a provitamin A molecular breeder. That NIFA sorghum project team, which involves expertise in testing under abiotic stress conditions in the Central Valley of California. That AI Institute project team, which again involves several breeding programs. A few members of our lab who are involved in the work that I described today. And funding from USAID, a Borlaug Feed the Future graduate research grant from USDA NIFA, this AI Institute here on campus, and finally to the UC Davis Department of Plant Sciences, in which I am based. Okay, thanks very much, and I will take a look at this Q&A and glad to answer any questions. Okay.
Thank you, Dr. Diepenbrock. Yeah, thank you very much for that awesome talk. Yeah, I think I can go ahead and read the questions and you could just answer them, if that works for you. Okay.
Absolutely.
Yeah. And the most amazing part of your talk was that you had so many national and international collaboration. That was pretty impressive. So I will go ahead with the first question. The first question is, how do you define levels and duration of water stress applied to the crops you use in your experiment? And how do you work the challenges of reproducibility of the stress imposition?
Sure. Yeah, that's a great question. So as is the next one too, Betsy. So yeah, so we end up in the crop growth modeling framework, we end up being able to quantify the water supply demand curve. So we can look at that through the season and see when that flowering stress was really imposed and to what extent. So that water, effectively the water supply demand stays at 1, a value of 1 where there's sufficient supply to meet the demand until it starts to drop. So that was an interesting way to look at that. In the case of pre-flowering drought, that ends up being quite a bit easier, given that in sorghum, at least, we define that as no watering from time of emergence to headed into the flowering stage. CIMMYT also has drought protocols that are able to be used, which were relied upon in Zimbabwe, where they look at maintaining soil, or excuse me, achieving soil dry down by a certain point in the season relative to flowering. So various ways. We have experts here as well who are able to assay soil moisture in various ways, soil neutron probes, some lower cost, less intrusive and easier to operate probes, etc. So real-time monitoring combined with like looking at actually how those dynamics are working in our model. Okay, and then great.
Yeah,
so
thank you.
Oh, sorry,
for
go ahead.
The answer. Oh, thank you for the answer. There was another question in the question and answer section, so I was just going to read it out.
Sure.
But were you going to do it yourself?
Oh, no, it's probably good if you read it.
Yeah, because the other participants are also listening, so I just wanted to make it loud and clear to everyone. So another question was, you mentioned that you are implementing the convolutional neural networks. Did you find that the CNNs outperform other neural network approaches?
Sure, that's a great question. So we are, how do I say? Our benchline or our baseline comparison at this point is not other neural nets. It's been other model types that might be more familiar to us as breeders. So yeah, part of our learning curve, you know, we have our typical baseline that we're trying to outperform with these new AI methods. My understanding is that some of the types of CNNs that we're implementing, convolutional neural nets, are beating other benchmarks like quite substantially. So it hasn't just been marginal gains, but some of these new types of deep learning and machine learning models are doing quite well in terms of substantial gains. So again, I'm not the expert on CNN method improvement and or implementation, but yeah, unfortunately so far I don't have a great comparison with other types of neural net. Although you could ask Vincent Colantonio, he might have thoughts on that point there at UF.
Well, thank you for that answer. There was another question, which is about biofortified orange maize. Was that maize engineered?
Oh, sure. Yeah, I didn't talk about the nutritional side today. That maize is not engineered. So that maize is developed through crosses. We do have extensive natural variation for carotenoid levels, including those with provitamin A activity in maize. So those were developed in efforts by CIMMYT and Harvest Plus. And yes, those are not engineered lines, thoroughly developed through crosses.
There was another question about the same maize, so I'll just go ahead and clear it out of the way. So are there any specific information on how that maize performed here in United States? That's the question.
Sure. Yeah. So again, in that case, we ended up effectively testing kind of parallel sets of germplasm that were relevant for each set of environments without having, yeah, I'm trying to think if there there's potentially some overlap, given that some of our panels were having some tropical subtropical representation, but I think in the case of the Zimbabwe panel that I mentioned, though, that would be almost entirely distinct from the diversity panels tested here here in the US as part of the work that I did. Again, there's probably others who are testing certain of these lines, but we did focus on how do I describe? So some of these collections are meant to represent global maize diversity and were selected accordingly from the available material. So it may be that those lines are related, but not exactly the same line selected in germplasm space. But yeah, that is something that we're working on in our projects going forward, is achieving partial overlap even across mega environments and looking at that performance as well. So that is an active area for us, but was not done in these past studies.
Thank you. There are a couple of more questions. So this one's, what type of equipment was used to collect the data to build the crop growth models in maize?
Oh, sure. Okay. Well, so the CGM-WGP project relied upon quite a bit of previous intensive physiological phenotyping. So in that case, they were looking at traits such as leaf area index, for which they could use various methods for that, say a leaf area meter or some kind of scanner. There end up being other traits in that model, like total number of leaves in maize. And then kind of the total, I guess the leaf area ends up being derived from total number of leaves and area of largest leaf. So there's just certain parameters in that model that ended up being trained over time. There was also fantastic work by Charlie and team before my time working on that team, looking at limited transpiration traits in maize. They've also had a really interesting study of silk, like issues with silk under drought. Silking being like the female, those strings that come out of the maize, there's one attached for each kernel. So they have a really neat paper out on that, and that type of information could potentially be integrated. So it's been, yeah, a lot of really careful studies by expert physiologists over the years in the case of that model.
Okay, so we have another question about the CGM-WGP model. This is, do you think it would be possible to apply this methodology using CGM-WGP in perennial crops? If yes, how challenging would that be?
Sure. Yeah, so I mean, I guess the previous speaker and I could have a discussion about this and see what we think, unless they were the one to ask the question. But I mean, it brings in, I think, a different set of considerations. You know, it seems, I'm not a perennial crop breeder, but there seem to be some different considerations with regards to main and interaction effects of G, E, and M in a perennial context. So I think which types of parameters you're modeling and what is your actual most of interest target of prediction could be really, you know, if you're interested in like current and subsequent cuttings, for instance, what is your final metric that you would like to be predicting, which intermediate emergent properties, so not just G, E, or M, but some combination of those. When I say emergent properties, it's kind of from like systems thinking, but of the system that is crop growth and development. Yeah, I think the targets could change, the ways that the models run could change, and again, I'm sure the DSAT developers would have thoughts on that part as well. But I mean, feasibly, the idea of the framework is generalizable, but there would probably need to be some changes. I also think, and yeah, I'll leave it there. But hypothetically, yes, that has not been tested, and of course, that's always in the details. Well, typically in the details of how these things work out.
Well, I just now got one more question about this model. So this is like, how and where do you see the CGM-WGP model being used in the future, especially in the face of climate change?
Sure. Yeah, again, I don't want to try to like oversell a model or a certain methodology or anything. It's a model that's in our toolkit and can be applied in certain contexts. I think a couple of areas where this method does show promise is, you know, once you've got the crop growth model reasonably well parameterized, it could be helpful in terms of not having to assay every physiological trait of interest in the entire population, but rather having really good priors, for instance, on the parents of that population, and then being able to, again, leverage this methodology alongside other genomic prediction strategies and see what gains there might be. We do see an advantage of CGM-WGP compared to WGP in situations of increasing G by E. In the case that we looked at, this meant decreasing whole season evapotranspiration. So the less whole season evapotranspiration that there was in a given environment, we ended up seeing CGM-WGP had a higher extent of predictive advantage. So again, in abiotic stress situations, these models may give the ability to have increased predictive ability might give you information that you could use to look at which individuals might be stress resilient and which traits might be underlying. You know, it's a hypothesis generator in a sense. And I saw Jeff Morris will speak tomorrow with regards to hypotheses as well. So yeah, it can help in predicting. It can help in generating hypotheses with regards to physiological mechanisms. And I thought of one more. This is interesting exercise on the fly. Oh, and it can provide a mechanism for integrating managed stress environment information, whereas before it might be really difficult to reconcile those data against those obtained in other environments.
Well, thank you for that detailed answer. There was one question about nutritional phenotyping specifically. So the question is, like, you mentioned about the nutritional phenotyping in your slides, but do you have any specific or standard method that you use in your lab for nutritional phenotyping? And the attendees would like to hear about it a little bit.
Okay, sure. Yes, so we do have multiple platforms, and it depends quite a bit on the trait. So for certain micronutrients, for instance, carotenoid compounds, HPLC or high performance liquid chromatography remains kind of the gold standard for phenotyping of those traits. If you want more of a like total composite measurement for that, or for phenolics, there are some existing colorimetric or 96-well plate-based assays that do exist that would use a plate reader, for instance, and read at a certain wavelength or set thereof. But yes, for purposes of improving the concentrations of specific nutritional compounds, we do end up looking at platforms such as HPLC. GCMS ends up being interesting for instance, for flavor aroma volatiles. Inductively coupled plasma mass spec ends up being really helpful for mineral nutrients. And then near-infrared spectroscopy we're using so far for macronutrients, so for that protein, fat, and starch, and maybe total phenolics. We'll see how that one goes.
Well, that were all the questions that I had for your talk. And then I once again want to say your talk was amazing. I personally enjoyed it a lot, and I'm sure that all my attendees feel the same. And your talk just made me feel like how technology is being used in agriculture and how we are moving forward. It's awesome.